James Dewey Watson was born on April 6, 1928, in Chicago into a middle-class family that valued education and intellectual achievement. His father, James D. Watson Sr., was a businessman who had struggled financially during the Great Depression but maintained a love of learning and a fascination with politics and current events. His mother, Jean Mitchell Watson, was the daughter of a Scottish immigrant tailor and had ambitions for her son that exceeded the modest circumstances of their South Side Chicago neighborhood. From the beginning, young James, called Jimmy by his family, was marked as special, as someone destined for something more than ordinary life. Jimmy was precocious, reading voraciously from an early age, showing an exceptional memory and a quick, sharp intelligence. But he wasn't the stereotypical bookish child. He was also a passionate bird watcher, spending hours observing and identifying species, developing the naturalist's eye for detail and pattern that would later serve him well in science. He was competitive, even combative in intellectual matters, always wanting to be the smartest person in the room, always ready to argue, always certain of his own correctness. These traits, the intellectual arrogance, the competitiveness, the unwillingness to defer to authority, would characterize his entire career. In 1943, at age 15, Watson enrolled at the University of Chicago through its special early entrance program for gifted students. The University of Chicago in the 1940s was an intellectual powerhouse, with a core curriculum emphasizing great books, rigorous thinking across disciplines, and debate. Watson thrived in this environment, though he later claimed he wasn't particularly serious about his studies, that he spent more time bird-watching than in the library. But he was clearly learning, absorbing ideas, developing the broad intellectual framework that would later allow him to think across disciplines. Watson's initial interest was zoology. He wanted to be an ornithologist, to study birds scientifically. But in 1946, at age 18, he read Erwin Schrödinger's What is Life?, the same book that had influenced so many physicists and chemists to turn to biology. Schrödinger's speculation that genes must be some kind of aperiodic crystal storing information in a molecular structure fascinated Watson. Here was a suggestion that the secret of life might be found in chemistry and physics, that understanding heredity might require understanding molecular structure. Watson's interests shifted decisively toward genetics. He completed his bachelor's degree in zoology in 1947 at age 19 and entered graduate school at Indiana University to study genetics under Salvador Luria and the Nobel laureate Hermann Muller. Indiana's genetics program was one of the best in the country, focused on bacteriophage genetics, studying viruses that infect bacteria. Luria became Watson's mentor, teaching him not just genetics but also the culture of molecular biology, the informal networks, the importance of conferences, the art of asking the right questions. Watson completed his PhD in 1950 at age 22 with a dissertation on the effects of X-rays on bacteriophage multiplication. It was solid work, but not groundbreaking. Watson was talented, clearly brilliant, but still searching for the big problem that would define his career. After his PhD, Watson did postdoctoral work in Copenhagen, supposedly studying bacterial metabolism, but he was bored and unproductive. Then, in the spring of 1951, Watson attended a conference in Naples where he heard a talk by Maurice Wilkins of King's College London. Wilkins showed an X-ray diffraction photograph of DNA, a fuzzy image, not particularly impressive to most of the audience, but to Watson, it was a revelation. If DNA could be crystallized and photographed with X-rays, its structure could potentially be determined. And if the structure could be determined, the secret of how genetic information is stored and replicated might be revealed. Watson knew immediately this was the problem he wanted to work on. This was his chance at greatness. In the fall of 1951, Watson finagled his way to Cambridge University's Cavendish Laboratory, ostensibly to work on protein structure, but really to position himself to work on DNA. At Cambridge, he met Francis Crick, a 35-year-old graduate student who hadn't yet completed his PhD. Crick was brilliant, talkative, ambitious, and frustrated that he hadn't yet made a significant contribution to science. The two men formed an instant intellectual partnership. Watson provided youth, energy, and knowledge of genetics. Crick provided physical intuition, knowledge of X-ray crystallography, and theoretical sophistication. Together, they would attempt to determine the structure of DNA, even though neither was officially supposed to be working on it. The challenge was formidable. DNA, deoxyribonucleic acid, was known to be important for heredity, probably the molecule that carried genetic information. But its structure was a mystery. The molecule was known to contain 4 bases: adenine, guanine, cytosine, thymine, sugar molecules, and phosphate groups, but how these fit together was unclear. The only way to determine the structure was through X-ray crystallography, taking X-ray photographs of crystallized DNA and using the diffraction patterns to deduce the molecular arrangement. But this required exceptional experimental skill, sophisticated mathematical analysis, and enormous patience. The best X-ray photographs of DNA were being produced at King's College London by Rosalind Franklin, a brilliant physical chemist who had been working on DNA structure since 1951. Franklin's photographs were extraordinarily clear and detailed, far better than anyone else's. She was also a rigorous, careful scientist who wanted to deduce the structure purely from the experimental data without making assumptions. But Franklin's relationship with her colleague Morris Wilkins was terrible. They barely spoke, competed for resources, and actively disliked each other. This dysfunction at King's would have profound consequences. Watson and Crick, meanwhile, were taking a different approach. Rather than generating their own X-ray data, they would build models, physical structures using metal plates and rods, trying different possible arrangements until they found one that fit the available data. It was a shortcut, potentially brilliant or potentially a waste of time. Their first serious attempt in late 1951 was a disaster. They proposed a triple helix structure with the phosphate groups in the center. When Franklin and Wilkins came to Cambridge to see the model, Franklin immediately pointed out that it contradicted basic chemistry. The phosphates would repel each other. Watson and Crick were embarrassed, and Sir Lawrence Bragg, head of the Cavendish, told them to stop working on DNA and leave it to the King's College group. But Watson and Crick couldn't stop thinking about DNA. Through 1952, they continued discussing it informally, reading papers, thinking about possible structures. Then, in January 1953, everything changed. Watson visited King's College and was shown, without Franklin's knowledge or permission, the X-ray photograph she called Photo 51. It was her clearest image of DNA, showing an unmistakable X pattern that indicated a helical structure. Watson also learned from Wilkins about Franklin's measurements of DNA's dimensions. This data, which Franklin had not yet published, gave Watson and Crick the key information they needed. Returning to Cambridge, Watson convinced Crick to resume model building despite Bragg's prohibition. Using Franklin's data, without her knowledge, chemical insights from the biochemist Erwin Chargaff, who had shown that in DNA, the amount of adenine always equals the amount of thymine, and guanine equals cytosine, and their own theoretical reasoning, Watson and Crick built model after model. The breakthrough came when Watson realized that the bases could pair in a specific way, adenine with thiamine, guanine with cytosine, forming the rungs of a ladder, with the sugar-phosphate backbones forming the sides. This pairing explained Chargaff's rules and suggested how DNA could replicate. The 2 strands could separate, and each could serve as a template for building a new complementary strand. By late February 1953, Watson and Crick had their structure: a double helix, with 2 antiparallel strands spiraling around each other, bases pointing inward and pairing specifically, phosphates on the outside. It was beautiful, elegant, and it immediately suggested how genetic information could be stored in the sequence of bases and replicated by strand separation and templating. On February 28, 1953, Crick walked into the Eagle pub in Cambridge and announced, with characteristic lack of modesty, "We have discovered the secret of life." They wrote up their result quickly and submitted it to Nature. The paper, published on April 25, 1953, was less than a page long, modestly titled "A Structure for Deoxyribonucleic Acid," and included the famous understated sentence, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." The paper appeared in the same issue of Nature alongside papers from Franklin and Wilkins, providing the X-ray evidence supporting the structure. It seemed like a collaborative triumph, 3 groups at 2 institutions coming together to solve one of biology's great mysteries. But the reality was more complicated. Franklin had been close to determining the structure herself, her data was the best, and she had been systematically working through possible structures. If Watson hadn't seen Photo 51 without her permission, if her data hadn't been shared without her knowledge, she might have gotten there first. Watson and Crick had used her data, but they had also provided the crucial theoretical insights: the base pairing, the antiparallel strands, the implications for replication. Who deserved credit? This question would haunt the discovery for decades. The double helix paper made Watson and Crick famous within the scientific community almost immediately. The structure was so elegant, so obviously right, and it explained so much: how genetic information could be stored in a linear sequence, how it could be replicated accurately, how mutations could arise from errors in replication or base pairing. Within a few years, the structure was universally accepted, and Watson and Crick were being hailed as the discoverers of the secret of life. But there were complications and controversies from the beginning. The role of Rosalind Franklin was ambiguous and uncomfortable. She had provided the critical X-ray data, but she hadn't determined the structure. Her paper in the same Nature issue supported the double helix, but didn't claim discovery. Over the following years, Franklin continued her work, moving from King's College to Birkbeck College and working on the tobacco mosaic virus, producing excellent structural work. But she was never fully credited for her contribution to DNA's structure during her lifetime. Then tragedy struck. In 1956, Rosalind Franklin was diagnosed with ovarian cancer, likely caused by her extensive exposure to X-rays during her years of crystallography work. She fought the disease bravely, continuing to work between treatments, but the cancer was aggressive. On April 16, 1958, at age 37, Franklin died. She never knew how history would remember her, never saw the recognition she deserved, never participated in the debates about credit and priority that would later surround the DNA discovery. The Nobel Prize for the discovery of DNA's structure was awarded in 1962 to Watson, Crick, and Wilkins. Franklin, having died 4 years earlier, was ineligible. The Nobel Prize cannot be awarded posthumously, and the prize can be shared by at most 3 people. This has led to endless debates. Would Franklin have been included if she had lived? Should the prize have been delayed until her contribution could be properly evaluated? Did Watson and Crick steal her work, or did they merely use publicly available data more cleverly than she did? Watson's own account of the discovery, published in 1968 as The Double Helix, made these controversies worse. The book was Watson's personal memoir of the race to discover DNA's structure, written in a breezy, informal style that made science accessible and exciting. It became a bestseller, bringing the story to millions of readers. But it also portrayed the scientists involved, especially Rosalind Franklin, in deeply problematic ways. Watson described Franklin, whom he called Rosie, a nickname she never used, as frumpy, difficult, unfeminine, and unable to interpret her own data properly. He described his annoyance at having to deal with her, his dismissal of her scientific abilities, and his focus on her appearance and manner rather than her work. When the book was about to be published, many of Watson's colleagues, including Crick, Wilkins, and Franklin's close friend Aaron Klug, protested the portrayal and tried to get Watson to revise it. Watson made some minor changes, but refused to substantially alter his account. The double helix created a firestorm. It was praised for its honesty, for showing that scientists are human beings with egos and ambitions, not dispassionate seekers of truth. But it was also criticized for its sexism, for its dismissiveness toward Franklin, for making the discovery seem like a lucky race rather than careful scientific work. The book transformed Franklin into a tragic figure, the brilliant woman scientist whose work was taken without credit, who died young without recognition, who was then insulted posthumously by one of the men who had benefited from her data. In later years, as the women's movement gained strength, and as historians examined the DNA discovery more carefully, Rosalind Franklin became a feminist icon, a symbol of how women scientists had been marginalized, denied credit, and erased from history. Her contribution was belatedly recognized. Buildings, institutes, and awards were named for her. Her scientific achievements beyond DNA, her work on viruses, on coal structure, were highlighted. The narrative shifted, from Watson and Crick as lone heroes to a more complex story involving multiple contributors, with Franklin as a victim of both sexism and bad luck. Watson's response to this reappraisal was defensive. He insisted he had told the truth in The Double Helix, that Franklin really had been difficult, that she hadn't understood her own data, that her contribution, while important, didn't warrant equal credit with his and Crick's. This defensiveness, combined with increasingly controversial public statements on other topics, would define Watson's later years and ultimately destroy his reputation. After the double helix discovery, Watson's career flourished. In 1953, at age 25, he was already famous. He spent time at Caltech working with the phage group, then returned to Cambridge, then joined the faculty at Harvard in 1956 at age 28. At Harvard, he continued working on nucleic acid structure and the genetic code, the problem of how the sequence of bases in DNA is translated into the sequence of amino acids in proteins. This work was important and influential, but it was clear that Watson's greatest achievement was behind him. The double helix would remain his defining contribution. But Watson had another ambition. He wanted to build an institution, to create a center for molecular biology that would attract the best scientists and make important discoveries. His vehicle was Cold Spring Harbor Laboratory, a small research facility on Long Island, New York, that had a distinguished history going back to the early 20th century, but had fallen on hard times by the 1960s. Watson became director of Cold Spring Harbor in 1968, and over the next 4 decades, he transformed it into one of the world's leading centers for genetics and molecular biology research. Watson's leadership style at Cold Spring Harbor was distinctive. He was demanding, sometimes abrasive, willing to take risks on young scientists with unconventional ideas. He raised enormous amounts of money from wealthy donors, built new facilities, expanded the research programs, and created an institution that consistently produced important work. Cold Spring Harbor became known for its summer courses, where young scientists came to learn new techniques, for its meetings bringing together researchers from around the world, and for its press publishing important books and journals in molecular biology. The work done at Cold Spring Harbor under Watson's leadership contributed significantly to the molecular biology revolution of the late 20th century. Research there helped elucidate the genetic code, understand gene regulation, develop recombinant DNA technology, and map cancer genes. Watson recruited brilliant scientists, gave them resources and freedom, and created an environment where important work could be done. By any measure, his tenure as director was successful. Cold Spring Harbor became a major scientific institution, and Watson deserved much of the credit. But Watson's success was accompanied by growing controversy over his public statements and attitudes. He developed a reputation for provocative, often offensive comments about women in science, about race and intelligence, about colleagues and competitors. Sometimes these comments seemed calculated to shock, to generate publicity, to position himself as a fearless truth-teller, willing to say what others wouldn't. But they also revealed attitudes that were, at best, outdated, and, at worst, deeply prejudiced. In the 1990s and 2000s, Watson became increasingly involved in the Human Genome Project, the international effort to sequence the entire human genome. As head of the National Institutes of Health's Genome Program from 1988 to 1992, Watson played a crucial role in getting the project funded and organized. He advocated for making genome data publicly available, for addressing ethical issues raised by genetic information, and for keeping the project on track. But he also clashed with NIH administrators over patents on gene sequences, resigned in frustration, and increasingly became a critic of how the Genome Project was being managed. Watson's vision for genomic medicine was largely realized. The human genome was sequenced by 2003, genome sequencing costs dropped dramatically, and genomic information became central to medicine and biology. But Watson himself was increasingly marginalized, his contributions acknowledged but his presence at scientific meetings increasingly uncomfortable due to his controversial statements. He was becoming a relic, a figure from the heroic age of molecular biology who hadn't adapted to the changing culture of science or the increased emphasis on diversity and inclusion. For decades, Watson had been making controversial statements that raised eyebrows but were often dismissed as eccentricity or provocative honesty. He'd said that women couldn't do science as well as men due to biological differences. He'd suggested that sunlight exposure might increase sexual drive in darker-skinned people. He'd made crude comments about the appearance of female students and colleagues. These statements were offensive, but they were often treated as gaffes from an older generation, as the politically incorrect musings of a brilliant but socially tone-deaf scientist. But in October 2007, Watson crossed a line from which there would be no return. In an interview with the Sunday Times of London, Watson made a series of statements about race and intelligence that caused an international outcry. He said he was inherently gloomy about the prospect of Africa, because all our social policies are based on the fact that their intelligence is the same as ours, whereas all the testing says not really. He suggested that while people wanted to believe all races have equal intelligence, evidence showed otherwise. When pressed, he said that people of African descent were genetically less intelligent than Europeans. The reaction was swift and devastating. Scientific institutions around the world condemned Watson's statements. The Cold Spring Harbor Laboratory Board of Trustees issued a statement disagreeing with his comments and affirming that no scientific evidence supports such views. Invitations to speak were canceled. The Science Museum in London canceled an appearance. Watson's decades of scientific achievement couldn't protect him from the consequences of his statements. He issued an apology, claiming he'd been misunderstood, that he didn't believe races differed in intelligence, that his remarks had been taken out of context. But the damage was done. Cold Spring Harbor Laboratory suspended Watson's administrative responsibilities, though he retained his title as chancellor. His reputation was in ruins. The man who had discovered the structure of DNA, who had helped launch the molecular biology revolution, who had built one of the world's great research institutions. This man was now radioactive, someone institutions couldn't be associated with without controversy and condemnation. Watson retreated from public life, but the controversies weren't over. In January 2019, a PBS documentary aired featuring Watson. In the film, Watson reiterated his views on race and intelligence, saying there was a genetic difference in IQ between blacks and whites, citing a difference on the average in IQ tests, and suggesting this was based on genetic differences. He had not changed his views, had not learned from the 2007 controversy, and remained committed to positions that the scientific community overwhelmingly rejected as racist and unsupported by evidence. The consequences this time were total. Cold Spring Harbor Laboratory stripped Watson of all honorary titles, Chancellor Emeritus, Professor Emeritus, the titles that marked his achievements and his long association with the institution he had built. The laboratory issued a statement saying Watson's remarks were reprehensible and unsupported by science, and that the lab unequivocally rejects these views. It was a complete repudiation. The institution Watson had led for 40 years was disowning him. The scientific basis for Watson's claims about race and intelligence is non-existent. While there are measurable differences in average IQ test scores between racial groups in the United States, the scientific consensus is that these differences reflect environmental, educational, and socioeconomic factors, not genetic differences in intelligence. Twin studies, adoption studies, and historical data showing IQ score improvements over time all indicate that intelligence is highly influenced by environment. The very concept of discrete biological races is scientifically problematic. Human genetic variation is continuous, not clustered into the racial categories we use socially. Watson, as a geneticist, should have known this. His training, his decades of experience studying genetics, his involvement in the Human Genome Project, all should have taught him that human genetic variation doesn't support racial hierarchies of intelligence. Yet he persisted in these views, suggesting that either he was willfully ignoring evidence or that his commitment to these ideas was based on something other than scientific reasoning. In 2020, Watson attempted to rehabilitate his finances by auctioning his Nobel Prize medal, the gold medal he'd received in 1962 for discovering DNA's structure. The medal sold for $1 million to Russian billionaire Alisher Usmanov, who immediately announced he would return it to Watson, saying the scientist's recent difficulties shouldn't diminish his earlier achievements. It was a generous gesture, but also a strange coda to Watson's career. The great scientist reduced to selling his most prestigious award, rescued by a billionaire's charity. How should we evaluate James Watson's legacy? On one side is an extraordinary scientific achievement. The discovery of DNA's double helix structure is one of the most important in the history of science. It revealed how genetic information is stored, how it's replicated, how evolution works at the molecular level. It launched the molecular biology revolution that transformed medicine, agriculture, and our understanding of life itself. Every advance in genetics since 1953, genetic engineering, genome sequencing, CRISPR gene editing, personalized medicine, builds on the foundation Watson and Crick laid. Watson's work establishing Cold Spring Harbor Laboratory as a premier research institution also matters. The institution has trained thousands of scientists, made countless discoveries, published important research. The summer courses, the meetings, the books, all have contributed significantly to advancing biology. Watson's vision, fundraising ability, and scientific judgment built something of lasting value that continues today despite his fall from grace. But on the other side is a troubling pattern of behavior and statements spanning decades. The sexism toward Rosalind Franklin and other women scientists. The racist statements about intelligence and race. The crude comments about appearance and sexuality. The refusal to learn or change despite criticism and evidence. This isn't just political incorrectness or old-fashioned attitudes. It represents a failure of scientific thinking, a willingness to make claims unsupported by evidence, a substitution of prejudice for reason. The contrast is striking. Watson was capable of extraordinary insight into molecular structure, able to see patterns in data that others missed, able to make logical leaps that revealed fundamental truths about genetics. Yet this same person was unable or unwilling to apply similar rigor to questions about human behavior, race, and gender. Why? Perhaps because examining his own prejudices would have required a kind of honesty he couldn't manage. Perhaps because the social and cultural biases he absorbed growing up in the 1930s and 1940s were too deeply ingrained to overcome. Perhaps because his early success made him arrogant, convinced of his own infallibility, even on topics where he had no expertise. There's also the question of how history should remember the DNA discovery itself. The traditional narrative, Watson and Crick as brilliant young scientists racing to uncover life's secret, has been complicated by our understanding of Rosalind Franklin's contribution. She provided crucial data without which the structure couldn't have been determined, yet she received no credit during her lifetime and no Nobel Prize. The discovery should be understood as a collaborative achievement involving multiple scientists at multiple institutions, with Franklin playing a central role. Watson and Crick's theoretical insights were crucial, but so was Franklin's experimental work. Some scientists and historians argue for separating Watson's scientific achievements from his personal failings, that we should honor the discovery of DNA's structure while condemning his racist and sexist statements that good science can come from flawed people. But others argue this separation is impossible, that Watson's attitudes affected his science, that his dismissiveness toward Franklin was rooted in the same sexism that would characterize his later statements, that his racial prejudices undermined his credibility as a scientist. The institutions associated with Watson have had to grapple with these questions practically. Cold Spring Harbor Laboratory honors his role in building the institution while distancing itself from his views. The Nobel Prize remains awarded to Watson, Crick, and Wilkins, as it should. They made a genuine discovery, whatever the complications around credit. But Watson's name has been removed from buildings, his honorary titles stripped, his presence at scientific meetings discouraged. He has been effectively erased from active participation in the scientific community he helped create. For young scientists learning about DNA, the story presents a moral complexity that previous generations didn't have to confront. They learn about the double helix, about Watson and Crick, about the brilliance of the discovery. But they also learn about Franklin, about Photo 51 obtained without permission, about sexism and racism, about the difference between scientific achievement and moral character. It's a more complicated story than the heroic narrative of Watson's youth, but it's also more honest, more reflective of how science actually works, through competition and collaboration, brilliance and prejudice, insight and exploitation. In the end, Watson's story is a cautionary tale about genius and its limits, about how scientific brilliance doesn't confer wisdom or moral insight, about how great achievements can be tarnished by personal failings. The double helix will remain one of science's great discoveries, but it will forever be associated with questions about collaboration, credit, sexism, and the extent to which we can separate scientific achievement from the character of scientists. Watson wanted to be remembered as the man who discovered the secret of life. Instead, he'll be remembered as the man who discovered the double helix, and then spent the rest of his life proving that scientific genius provides no immunity to prejudice, no guarantee of decency, and no escape from the consequences of one's words and actions. The structure stands, two antiparallel strands, bases paired in the middle, spiraling elegantly, containing in its sequence the information that makes us human. But the story of its discovery, like the man who claimed credit for it, is more twisted, more complicated, more troubling than the simple beauty of the molecule itself. That complexity, that acknowledgement that great science can emerge from morally questionable behavior, that brilliant people can hold reprehensible views, that discovery doesn't cleanse character. These uncomfortable truths are part of Watson's legacy too, as permanent as the double helix itself.